Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about selectable markers. Okay. In the previous video lecture we talked about the vectors, that what are the vector, what are the main properties of the vector. Okay, and we also talked about the types of the vector on the computer screen. Now this is another video lecture which is also regarded to recombinant DNA technology, and in this video lecture we are going to talk about selectable marker, and we are going to talk about the screening process. Okay, that how we can identify that our gene of interest is inserted or no. We call that also screening. Now before going to explain selectable marker, you guys need to understand the basic of recombinant DNA technology, and you guys need to understand that what are the vector and types of the vector. If you missed that video lecture, it is present into my channel. You can watch it, or don't worry, I will give the link in the description box. So let me erase this one portion and let me start. So let me write selectable, or we can say screening process or screening. Now first of all let me explain that what are the screening. If we talked about the definition of the screening on molecular level, or if we talk the definition of the screening in case of recombinant DNA technology, so it is that technique or that method due to which we can identify that whether our gene of interest is inserted into a vector or not, which contain our gene of interest is inserted into a bacterial cell or not. That's called as screening. Now then what are the selectable markers? If we talked about the selectable marker, so selectable markers are actually, these are the artificial gene sequences. Artificial gene sequences. Now whenever we talked about the marker, so marker doesn't mean that we have just like that marker. No, whenever we talked about the markers, so marker means that these are actually artificial gene sequences. Okay, in case of recombinant DNA technology, let me once again tell you, in case of recombinant DNA technology, marker means artificial gene sequences. What are the selectable markers? Selectable markers, these are those genes which we select for those purpose to identify our gene of interest, or to identify the vector which contain our gene of interest. That's why we call it selectable marker, to select those genes which have the ability to identify our gene of interest inside a vector. Okay, if we explain the selectable marker, so what actually happened, let's say we have our gene, let's say we have a vector here. Okay, so let's say these are the plasmid, because plasmid are so common. We can use plasmid in recombinant DNA technology so commonly. Now let's say we take a vector and we cleave it with restriction enzyme, and we also take our gene of interest and we also cleave it with the same restriction enzyme which we cleave the vector. Now once we cleave, then we insert our gene of interest here, let's say. Now after that, what actually we are going to do, we just insert this vector. This is now called as recombinant DNA. Why? Because in this case we combine our gene of interest into vector. After that, once we get the recombinant DNA, so our next step is we should insert this recombinant DNA into a bacterial host cell. Okay, now once the bacterial cell get the recombinant DNA technology, so after that this cell will start dividing. Now whenever, let's say this bacterial cell contain our gene of interest also, so this is a vector and this is our gene of interest. Okay, so now whenever the bacterial cell is growing and dividing, so our gene of interest is also growing and dividing. It is replicated, translated and transcribed and translated. Okay, so So let's suppose the phenomena that actually happen. Now this recombinant DNA or the plasmid is not directly internalized, or they are not directly transformed inside a bacterial cell. So when we want to transfer this recombinant DNA to the bacterial cell, so we need a process we call a transformation. And all of you better know that during transformation, what actually we do, we treat this bacterial cell with calcium chloride, or we provide we provide small electric current to the bacterial cell, due to which a pores is created and the bacterial cell can forcefully take the cell. Okay, they forcefully take the plasmid which contain our target DNA. Now once they take the plasmid, so then such bacteria are then divided, growing, due to which our target DNA is also growing and dividing. Okay, so that is called as transformation. Due to transformation, we can transfer our gene of interest, or we can transfer the vector by treating the bacteria with calcium chloride. But what actually happen? There is a problem. The main problem is that we can't see that whether the process is going on or not. It is, let's suppose that the same process are happening in a test tube, the same process are happening in a pre-prepared dishes. So all these processes are happening in a test tube, all these processes are happening in a petri plates. Okay, all these processes are happening, but we don't know that because all the cell is too much small. If the cell is too much small, so we can't see it, the plasmid is also small, we can't see it. So how we can identify what is actually going on, whether our gene of interest is inserted into a vector, whether this recombined vector, means whether this recombinant DNA is inserted into a bacterial cell, whether the bacterial cell is dividing or growing or not, whether our gene of interest is present inside a bacterial cell. We don't know about that. For this purpose, we use the selectable markers. We use the selectable markers to ensure, to check that whether our gene of interest is present or not. Okay, so there are many types of the selectable marker, or we can say there are many examples of the selectable markers. We have, sometimes we use antibiotic resistance gene. Use antibiotic resistance genes. We use fluorescence, color fluorescence. We use tag sequences. Okay, so these are the selectable markers, and these are the main things due to which we can identify our gene of interest. Okay, now by using each selectable marker, we can easily identify. Now what actually happen? Let's say we have a vector. Now if we have a vector, so at somewhere we treat the vector with restriction enzyme and we add our selectable markers here. So at that specific position, the selectable marker is added. And here we add, let's suppose, antibiotic resistance genes. And in this case, we add here ampicillin resistance genes. Now, once we add ampicillin resistance genes, then what actually happen? The cells, this means this vector contain the antibiotic resistance genes, as well as this vector also contain our gene of interest. So let's say this is this blue one representing the gene of interest. Now once we internalize this vector inside the bacterial host cell or inside the bacterial cell, then this cell will divide it. And let's say we also grow these cells into a petri plate. Let's say this is a petri plate. But remember, what actually we are going to do? We should make agar plate having ampicillin antibiotic. Now once we prepare ampicillin antibiotic agar plates, so then we grow our bacterial cell. So those bacteria, now remember, now here you will get the advantages, or you will get that how the selectable marker are working. Those bacteria which have the resistance genes, ampicillin resistance genes, so each bacteria will grow. And this is called the screening, and we can identify that our gene of interest is present and our vector is present here, which contain our gene of interest. So those bacteria will grow. Why it is grow? Because each bacteria contain the selectable markers, which is the ampicillin resistance genes. So the bacteria will grow, And those bacteria which don't have the ampicillin resistance genes, as well as those bacteria which don't have selectable markers, so these bacteria will not grow, because these bacteria don't have the resistance genes, so they will not protect their self from the antibiotic. So an antibiotic will kill each of the bacteria. So it shows the bacteria which grow, it shows that your gene of interest is present and your cells are growing on. That is called as screening, and that is called the selectable marker, and we call that positive selectable marker. Now, positive selectable marker, let me once again tell you, in this case we add the sequences, those kind of sequences due to which the bacterial cell are growing into a plates, into antibiotic plates. Okay, we call that positive selectable markers, due to which we can identify that our gene of interest is present inside the bacterial cell. We have another type of selectable marker, we call that negative selectable marker. Negative selectable marker. Now, what are the negative selectable? It is opposite to the positive. In negative selectable marker, the bacterial cell are not grow. When the bacterial cell are not grow, just think. Let's say this is not an ampicillin antibiotic, or let's say this is the ampicillin antibiotic resistant ampicillin antibiotic agar plates. Now, when we grow our bacteria here, so the bacteria will not grow. When we using negative selectable marker, the bacteria will not grow. So those bacteria which grow, we just think that these bacteria are not our these are not those bacteria which have our gene of interest. These are not those bacteria. So those bacteria which don't grow, we just identify that the bacteria which don't grow on the plate, these are actually those bacteria which contain our gene of interest. Okay, that is called as negative selectable marker. Usually we use positive selectable marker to visualize our gene of interest. Okay, now in this case, if we use selectable marker, so we can also know that our genetic expression, or we can measure outside, we can measure the genetic expression outside. Okay, so due to the selectable marker, we can measure our genetic expression outside. Now, that's all about the selectable marker, and that's how we can doing the screening. Although there are many types of the screening, we have white blue screening, we have some other types of the screening. Inshallah, in the next video lecture, we are going to talk about the screening method. Although we talked here, the screening means this is how the screening, this is how we can check that whether our gene of interest are present inside the bacterial cell or not. This is how the screening. But inshallah, in the next video lecture, there are many types of the screening. We are going to talk about the screening in detail. Okay, I hope you get an idea about the selectable marker or the screening. If you like this video lecture, so make sure to hit the like button, share this video, and if you have any kind of question in your mind, you can write it into a comment. Inshallah, I will give you answer as soon as possible. So like this video, share this video, and subscribe the channel to get more interesting videos like that. Thank you so much for watching.